Hello, good evening, everyone. So, this is Kanika Gupta, a PhD scholar from IIT Delhi, and your TA for the session. Today, we'll be discussing about week 6 lectures doubts, and this session will go as it has always been. First, we'll just have a recap of what we learned in week 6, and then we'll discuss about the questions of the assignment. Okay, so let's proceed towards our session. In this week, we learned about transcription. What is transcription? If you remember, in week third or fourth, I'm not very sure. We learned about central dogma of biology. So, in central dogma of biology, there were these processes. First of all, replication, then transcription, then translation. So here, replication we have discussed in the last week. In this, sorry, in last to last week, like week 5 and week 6 we have learned about transcription, what is transcription and how it is done. So we'll have a recap of what transcription is. So transcription is basically, as you all know, transcription, if we'll generally say it, it is the formation of RNA from DNA. Transcription is simply the formation of RNA from DNA. But if we'll go into the detail, what are the different things that are involved in transcription, what are different units, what are the different regions that are involved in transcription, is what will make the detail of the process. So every cell mainly contains three types of RNA. Basically, the transfer RNA, which is called as the tRNA, ribosomal RNA called as the rRNA, messenger RNA called as the mRNA. So synthesis of RNA from DNA templates with the help of DNA-dependent RNA polymerases is known as transcription. So what is transcription? Synthesis of RNA from DNA. Formation of DNA from DNA is replication. Formation of RNA from DNA is transcription, and formation of RNA from RNA, again replication. Okay, so synthesis of RNA from DNA templates with the help of DNA dependent RNA polymerases is known as transcription. Okay, and when we'll say the formation of DNA from RNA, that is called as the reverse transcription, because the process is getting reversed. Now RNA is not getting formed from DNA, it is the DNA which is forming because of the RNA. RNA is the template and DNA will be formed according to that template. So that will be called as reverse transcription. So synthesis of RNA from DNA templates with the help of DNA dependent RNA polymerases is known as transcription. It occurs unidirectionally, in which chain is synthesized in 5 prime to 3 prime direction. Okay, and the segment which is transcribed from DNA is known as what? Transcription unit. That segment will be called as transcription unit. In eukaryotes, it is the monocystronic transcription unit occur, in which coding sequence presents only for one polypeptide, while in case of prokaryotes it is polycystronic. Transcription unit generally have coding sequences for more than one polypeptide. Okay, so it is monocystronic in case of eukaryotes, it is polycystronic in case of prokaryotes. This is the thing we are talking about, transcription unit. Okay, so what happens in transcription? The starting point is, it is the first base pair from where transcription starts, and it is called as the start site. Okay, so RNA polymerase moves from start point along with the template, synthesize the RNA up to the terminator sequence. The sequence where it is marked that it is a terminator sequence, now you have to stop the transcription there, then the transcription will stop and it will start from the start point, the start site. Okay, so upstream position क्या होती है? It is the known template nucleotide is in 5 prime end or minus direction. Basically, when we are saying that transcription is occurring from 5 prime to 3 prime, so anything above, when the transcription is starting, the start point, anything above that start point is called as upstream. Anything before that start point is called as the upstream or minus direction. Okay, downstream, what is downstream? It is the nucleotide in 3 prime end or plus direction or sequence after start point. So anything which is after start point and in the 3 prime direction is called as the downstream direction. So this is basically the direction we are talking about, because these terms are generally used to define different kind of promoter sequencing and other supporting sequences, regulatory sequences that are present in a transcription unit, so that the transcription is successful. Okay. So DNA is a double stranded structure. During transcription, only one strand is transcribed, so that transcribed sequence is identical with one strand of DNA because it is complementary. The strand to which this RNA sequence is exactly similar is called as coding or sense strand, and other complementary strand is known as the template or anti sense strand, because उसके complementary तो हमारी जो transcription है वो हो रही है. So the sequence to which the RNA sequence is completely similar is called as the coding sequence or sense strand. And the sequence which is used as template is called as the antisense strand. Okay. Okay, so what is the difference between a prokaryotic and eukaryotic transcription? So prokaryotic transcription is a polycistronic type of transcription, while eukaryotic is monocystronic. The prokaryotic transcription occurs in cytoplasm, while eukaryotic in nucleus. Prokaryotic coupled transcription, translation process occurs simultaneously. Basically, what happens in prokaryotes, as soon as the mRNA starts getting formed, ribosomes will also come there and it will get attached to the formed part of mRNA, and it will together start forming the protein, the translation process. So that is coupled in case of the prokaryotes, while this is not the case in case of eukaryotes. Why it is not the case, you will come to learn in the lecture when we'll move ahead, because there are some kind of the post transcription modification that occurs in the mRNA of eukaryotes. So that's why it is not coupled in case of eukaryotes, while it is coupled in case of the prokaryotes. Okay. In prokaryotes, single type of RNA polymerase is required for synthesis of all types of RNA, while there are 3 types of RNA polymerases that are required for all types of RNA in case of eukaryotes. Okay. In prokaryotes, no need of any transcription factor for initiation, while in eukaryotes, it requires transcription factors for initiation. Those transcription factors actually play the regulatory role. They'll tell us कि now the transcription needs to be started. Now it needs to be moved ahead, and many proteins that will get attached to that transcription unit to make it go move smoothly. So transcription factor is a very important protein present inside the cell of eukaryotes to uh, make a systematic way out of it. In prokaryotes, RNA polymerases are made up by 5 subunits, while in case of eukaryotes, RNA polymerase are made up by 10 to 15 subunits. So it is a big compound, big protein, while in case of eukaryotes, it is comparatively smaller. Okay, next is transcription in prokaryotes. How this happens, we'll not go into the detail of the how transcription in prokaryotes happens, but we'll just have an overview that how it happens. Sorry. So transcription in prokaryotes, the enzyme involved is RNA polymerase. As I told you, it is made up of 5 subunits. So in prokaryotes, single type of RNA polymerase is present, which is responsible for synthesis of all types of RNA. Bacterial RNA polymerase is named as holoenzyme, is a multi-subunit protein which contains 5 subunits. Okay, alpha, beta, beta prime, alpha, और ये है डेल्टा. Okay, so alpha is responsible for assembly of the core enzyme. Beta and beta prime performs all the enzymatic and catalytic functions. Delta recognizes the promoter sequences. Okay, it will recognize the promoter sequences. So alpha, 2 subunits of alpha, beta and beta prime forms the core enzyme, and this delta together makes this enzyme complete. Sorry, sigma. I'm very sorry, it is not delta, it is sigma. So this sigma makes the enzyme complete. When this sigma gets attached to the core enzyme, then it makes the enzyme complete, and then that enzyme will be called as holoenzyme, because now the subunits have attached properly and now the enzyme is complete to function. Now, prokaryotic promoter. What is the structure of prokaryotic promoter? So, promoter typically consists of 40 base pair region located near to 5 prime end site of transcription start site. ओके, स्टार्ट साइट का जो एंड साइट है, एक मिनट, हाँ, so promoter typically consists of 40 base pair region located near to 5 prime end site of transcription start site, and promoter region consists of two 6 base pair consensus sequences elements, Pribnow box or Tata box, and a minus 35 region. That means an upstream region में minus 35 position पे, and the Pribna box 10 base pair upstream of the start point is a consensus sequence, TAT. So this minus 35 region has consensus sequence TTGACA. So basically there are two sequences at minus 35 position, and 6 base pair sequence of this Pribnow box, Strata box, and sorry, at 35 position, and minus 10 base pair upstream of start site. Basically, Tata box is first present first, then this TAAT sequence, and then the start site. So this is how the arrangement of the prokaryotic promoter is. Okay, so transcription in prokaryotes, what are the stages in which the transcription will happen? So transcription occurs in four stages. First, the template binding, then the chain initiation. Template binding means here is this template will be attached with an RNA polymerase. Sorry, with an RNA polymerase, a primer will be added there, and then the sequence will start forming the transcription. Then the chain initiation will happen. Now the RNA polymerase has done the initial recognition part and all. Now it is ready to synthesize the RNA. Then the chain initiation will happen. Then after the chain elongation will happen, the complete transcription will happen. And then when the RNA polymerase reaches the a termination site, then the termination of transcription will happen. So in the first step, what happens is binding of RNA polymerase to template DNA and chain initiation. So DNA duplex should be opened so that RNA polymerase can approach to single stranded DNA template. Efficiency of initiation is inversely proportional to the melting temperature. As much as high is the melting temperature, that means more energy is required to open up the DNA. So less efficiency will be there, because maximum of the energy will be spent in opening of the DNA. So high will be the melting temperature, lower will be the efficiency, and if efficiency of initiation, and if the melting temperature is low, then the efficiency of initiation will be higher. Okay, so obviously the AT rich region will have a lower TM, so it will have higher energy, higher efficiency of initiation compared to the GC rich region. Okay, RNA polymerase also have a sigma factor, which recognizes the promoter sequence at which the RNA polymerase holoenzyme binds and forms a complex which is known as the closed complex. Okay, in fact, sigma's factor is released when chain reaches nearly up to 10 bases, leaving core enzyme for further elongation. So it is all a regulated process, so that nothing can go wrong in the initiation. If an RNA replication, or we can say transcription part, goes wrong, then obviously a protein will be faulty, and that faulty protein will be very dangerous for the cell. So that's why this process is very controlled and very regulated. So we are not going into the detail of how the chain elongation will happen, how the chain initiation has happened. Finally, you guys must have read that in the videos that sir had provided. We are directly coming to the chain termination part. When the RNA polymerase stops adding nucleotide at RNA chain, it releases a completed product, and RNA chain gets free from the terminator sequence. Okay, so it releases the final product, and RNA chain will now get released from the wherever it got terminated, and during termination all the hydrogen bonds break down, which hold RNA and DNA hybrid together. And when RNA chain is separated, DNA again reform the duplex. So DNA will again come to its original position, RNA will be detached from this duplex, because now the termination has the transcription has terminated. So site at which site enzyme stops adding nucleotides is known as the chain termination site. Now the termination is of two types: either it is an intrinsic termination or it can be a row factor dependent termination in case of the prokaryotes. Okay, so it can be of two types: the intrinsic or the factor dependent termination. Now let's go into the detail what are these intrinsic and what are the row factor dependent. So in case of the intrinsic termination, what happens is, in this mechanism of termination, row factor is not required and termination depends upon RNA product. It requires GC rich hairpin structure is followed by 7 U residues. So it is just inbuilt in RNA only. RNA DNA hybrid requires forces for holding the elongation complex together. Thus, when the hybrid gets detached, it collapses the elongation complex, which causes the termination. In this type of termination, the dissociation of polymerase occurs by destabilizing the attachment of growing chain to the template. Okay, and during this process, hairpin structure is formed by the transcription via complementary base pairing. So hairpin structure will be formed in the RNA only. It will make a double strand with its own strand. It will fold on itself and it will make a hairpin like structure, which leads to the termination here. Okay, via complementary base pairing. And it includes palindromic sequences. This stem loop structure includes GC rich region followed by U rich region. So this includes the GC rich region that comes after the 7 U residues of this uracil, and then after this GC rich region comes, which helps in the formation of this hairpin structure and leads to the termination. While in case of this sorry, it is just the intrinsic transmission. So different steps are here: two inverted repeats GCC, GGG, G triple C, GC are present in the DNA template, which is transcribed. Nearly 6 adenine residues followed by the second inverted repeat this. So basically 6 adenine residues comes in between, so that this can get enough room to make a hairpin structure and get complementary binding with the already present G triple C GC. Okay, now the inverted repeats are forming a hairpin structure which pauses the polymerase, and due to the formation of stem loop structure, it is also called as the stem loop structure, the AU bonds gets breakdown, leads to a termination, and RNA molecules get separated. Okay, so this is how it happens. If you can see it from the structure also, from this pictures also. Now the row dependent termination. What happens in row dependent termination? In this type of termination requires a row protein. Row is an ATP dependent helicase that disrupts RNA DNA hybrid. It is an essential protein which causes transcription termination. Rho protein is an hexamer ATP dependent helicase and its subunit contain RNA binding and ATP hydrolysis domain. So it is a multi-unit hexamer ATP dependent protein. The Rho protein firstly binds to the sequence which is present at upstream of the termination site, that means in the 5' direction. And these sites are called rut sites, RUT, rut sites. These sites are rich in C residues. Rho factor followed to RNA polymerase until it do not catch RNA polymerase. Rho follow the RNA polymerase by its helicase activity, which is driven by ATP hydrolysis. When RNA polymerase reached at terminate site, the Rho protein freezes the structure of RNA polymerase, and when Rho factor collapses with enzyme which causes the termination and new RNA chain gets release. So basically, rho factor chases the RNA polymerase, and as soon as the RNA polymerase reaches the terminator sequence, this rho factor will freeze the RNA polymerase, and the both of them frozen structures will fall off from the hybrid structure, and new RNA will be released from there. ठीक है. So RNA polymerases, if we'll go into the this was all about the transcription in prokaryotes. Now we'll see about the eukaryotes. So first of all, as we all read in the first third slides, that the RNA polymerases in case of eukaryotes are of three types. In prokaryotes it was of one type, but in eukaryotes it is of three types. The RNA polymerase of mitochondria and chloroplast are similar like bacteria, and all eukaryotic RNA polymerases are multi-subunit proteins which contain three different types of RNA polymerases, which are RNA polymerase 1. This synthesizes rRNA, which is resistant to amytin. Amytin is a poison released by a fungus, and it occurs in nucleoli. RNA polymerase 2, it leads to a synthesis of mRNA very sensitive to amytin, and occurs in nucleoplasm, basically nucleus. RNA polymerase 3, it helps in the synthesis of tRNA. It is less sensitive to amytin, and it is present in the nucleoplasm again. Okay, so promoter of eukaryotes, each promoter contains some specific sequences which get recognized by transcription factor. Now here the transcription factor will come into action. In prokaryotes there was no transcription factor involved, but in eukaryotes there is a transcription factor involved. So eukaryotic promoter has longer region than prokaryotic promoter because it contains all those sequences which are important regarding to initiation. It includes core promoter element at which RNA polymerase get attached and form the initiation complex. So it is longer than the prokaryotic promoter, and this contains all of the sequences which is important for the initiation. Okay. And also for the efficient transcription, it requires an upstream promoter element, an element which is present in eukaryotes only, which is present upstream to the promoter, which are basically GC rich region and at which transcription factors actually bind. Okay, so in case of eukaryotes, the initiation process and the obviously the promoter is different from prokaryotes. Okay, so transcription initiation by RNA polymerase 2 happens in case of eukaryotes. RNA polymerase 2 will help in the formation of mRNA. Sorry. So eukaryotic mRNA transcription requires initiation complex which consists general transcription factors and mediators. So these are all the GTFs, or we can say general transcription factors as listed. सबसे पहले ट्रांसक्रिप्शन फैक्टर 2D, it is further divided into two types. Tata binding, it has two parts basically, the Tata binding protein and TBP associated factors, so TAFs. What happens is it recognizes the core promoter, the Tata box, and recognized to core promoter non-Tata box. So basically this TBP and TAFs are actually the one which is recognizing the Tata box, the Tata binding protein, and the TBP associated factors, the Tata binding protein associated factors will help in this kind of the recognition. They will not directly bind to Tata box, but they will help in this recognition and binding. Okay, so TF2A is the next transcription factor which stabilizes the TBPs and TAF binding. So when the binding happens, the TF2A will help in the stabilization of this interaction. TF2B, it helps in the RNA polymerase 2, and TF2F recruitment also helps in start site selection. TF2F, what does it do? It helps the RNA polymerase in promoter binding. So basically all of these transcription factors together come into action to help in the formation of an RNA polymerase and promoter sequence binding complex. TF2E, what it helps in? It helps in the TF2H recruitment, modulation of TF2H helicase, ATPase, kinase activities. And TF2H, what does it do? It helps in the promoter melting with helicase activities, promoter clearance by phosphorylation. So this is how all of these factors are involved, and as mentioned, they come in this series only: 1, 2, 3, 4, 5, 6. Okay, so transcription factor initiation by RNA polymerase. These transcription factors are sequentially bind to Tata box DNA to form a pre-initiation complex, and at last, when TF2H get binds, it phosphorylates RNA polymerase 2 basically to initiate the transcription in the presence of ATP. So it is an energy requiring process. Now we are again not going into the detail of the chain initiation, elongation and all. We are directly coming to the termination part. So RNA polymerase 2 transcription genes may continue up to hundreds or even thousands of nucleotides beyond the end of a coding sequence because of the multiple kind of the regulatory sequences being present there. So then the cleavage of RNA strand occurs by a complex which appears to associate with polymerase. Cleavage of RNA is coupled with the termination process and it occurs at same consensus sequences. The polyadenylation of mature polymerase 2 mRNA occurs at the 3' end. Basically, a poly A tail addition happens there. So this poly A tail will get attached at the 3' end, and this process is followed by the cleavage and termination. Both processes, polyadenylation and termination, occurs at the same consensus sequences, and both these processes are interdependent. Okay, so now if we'll go into the detail of what kind of a polymerase requires what kind of a termination. So polymerase 1 genes requires row dependent termination. Just a moment, guys. So polymerase 1 requires row dependent termination. Polymerase 3 does not require row factor for termination. While polymerase 2 genes, it is more complex. Polymerase 3 termination generally coupled with an RNA processing event in which basically polymerase 2, it must be written writing error, in which 3' end of the transcript undergoes cleavage and polyadenylation. Okay, so it is the addition of the poly A tail adenyl group. So transcription in eukaryotes, the actual termination. So poly A dependent termination is the this type of termination are basically coupled with the RNA maturation process in which the 3' end of the nascent RNA undergoes polyadenylation and cleavage, and these 3' end processing reactions are carried out in two steps: transcription of poly A tail followed by cleavage of the nascent transcript, and then the upstream product is polyadenylated and the downstream product is degraded. Basically 3' end processing starts when the cis acting element in poly A site of nascent RNA transcript is recognized by the binding factor. So there are many factors involved in the whole process. When these factors bind at 3' end, it forms a very complex structure which results in high shear forces. Consequently, processing slow down, which causes the disruption of polymerase 2 and DNA RNA hybrid complex. So this is was the basically the transcription in eukaryotes. This was transcription in eukaryotes. Now we'll move towards the post-transcriptional modification. So as I've already told you, in case of prokaryotes, there is no such kind of post-transcriptional modification occurs, but in case of the eukaryotes, it does happens. It is the addition of a poly A tail on the 3' and GC cap. Basically, capping occurs at the 5' end. So there are basically these two additions, these two post transcription modification, and third is the splicing out of the introns, because when RNA is getting formed, this mRNA is getting formed, it will transcribe the whole gene. And in eukaryotes, we all know in eukaryotic gene we have introns present in them, so they must be removed from the messenger RNA so that the protein won't be affected. So that splicing out of introns also happens as a post transcriptional modification in case of mRNA in eukaryotes. So intron splicing, how does it happen? Introns are non-coding nucleotide sequence within a gene that don't code for a protein and don't appear in the final mRNA molecule and are removed by splicing. Protein coding sequence of a gene known as exons, which are interrupted by introns. So the vast majority of eukaryotic genes are interrupted with non-coding region introns, which needs to be spliced out. However, histone protein coding genes in vertebrates is one among few exceptions. Okay. So histone protein coding gene is one among the few exceptions which does not generally have this non-coding region. It does not have the non-coding region. So the occurrence of introns varies in eukaryote species. Some E species lacks introns and many genes in eukaryotes carry a dozen of them. Few bacterial and archaeal genes also have introns, and introns can vary in length from 50 to 20,000 nucleotides. इंट्रॉन्स की लेंथ जो है वो वेरी करती हैं, एंड इन हायर एनिमल्स एज ह्यूमंस इंट्रॉन्स आर मोर देन एक्सॉन्स. ठीक है. So there are four classes of introns. Introns are further divided into classes: group 1, group 2, then spliceosomal introns, and then the introns that require ATP for splicing. ठीक है. Group 1 and group 2 are both self splicing introns and does not involve any protein या enzymes. ठीक है. Splicing mechanism of group 1 and group 2, basically splicing mechanism of both group 1 and group 2 involve similar steps of two transesterification reactions. So there happens two transesterification reactions, sorry, in which a ribose 2' or 3' hydroxyl groups make a nucleophilic attack on the phosphorus and a new phosphodiester bond is formed at the expense of the old one. Okay, so this is how this self splicing happens. Mechanism of these groups differ in nucleophile which is used. Group 1 uses 3' hydroxyl group of guanine nucleotide as nucleophile, while group 1 introns are found in some nuclear mitochondrial and chloroplasts that code for rRNAs, mRNAs and tRNAs. ठीक है. While in group 2 a lariat formation will occur. Now alternative splicing, how does it happen? So alternative splicing is a method substantially used for many mammalian genes, can result in multiple products that vary structurally and functionally from the same primary transcript. So alternative splicing method is generally used in eukaryotes in case of the mammalian genes. Sometimes alternate splicing is unregulated phenomena, while in some it is strictly regulated. One of the best example of regulated alternative splicing occurs in sex determination in Drosophila. In Drosophila, three genes are involved in sex determination: sex lethal gene SXL, transformer gene TRA, double sex gene DSX. So these three genes are formed from the single mRNA transcript just by the alternative splicing of that single transcript. Okay, so due to alternative splicing, functional genes are produced in females and non-functional genes are produced in males. Alternative splicing occurs using two mechanisms. One, two poly A or cleavage site are available in primary transcript, and cleavage occurs at either site, resulting in two different products. Such mechanism is followed by variable domains of immunoglobulin heavy chains and their diversity due to this mechanism, because obviously the antibodies must be knowing the diversity, must be having the diversity so that they can tackle multiple organisms. Okay, so this was what alternative splicing is. Now the post transcription modification, the process when the rRNA is formed, then it also faces some kind of the post transcriptional modification. So the steps in the rRNA processing are: eukaryotics have 80s ribosomes, and prokaryotes have 70s ribosomes. So ribosomal RNAs are transcribed as long precursors sequence, which is then modified at specific bases and cleaved to give mature products. In both bacterial and eukaryotic, rRNA processing involves two basic steps of cleavage and the base modification. So, a precursor in bacteria is a 30S rRNA which modified cleaved to give 23S rRNA, 16S rRNA, and 5S rRNA. So, this is the single rRNA which is then further modified and cleaved to give three different types of rRNA. And some tRNA segments in between are also there sometimes. The 30S pre-RNA transcripts consist of 16S rRNA sequence followed by spacer which may have tRNA. Okay, sequence in some cases, and then there is a 23S rRNA sequence followed by a 5S rRNA sequence near to 3 prime end. At times there is one more tRNA sequence after 5S rRNA sequence and 3 prime end. There are 7 different genes of rRNA in E. coli. They are essentially similar in sequence of RNA segment but differ with the number and sequence of the tRNA segments. So maturation process involves the methylation of 30S RNA precursor at specific sites so that it won't be degraded, occurring at 2' hydroxyl group of the bases. Some bases such as uridine is modified to pseudouridine or dihydrouridine. These are all mechanisms to protect it from getting degraded. Okay. Further cleavage process is carried out using enzyme RNase 3, RNase P and RNase E at sites 1, 2 and 3 respectively, and then the further RNAs are formed. ठीक है. So now the RNA processing in case of the vertebrates. How does it occur in eukaryotes? Nucleus is the center of processing ribosomal RNA. A 45S precursor RNA. There was 30S rRNA, here 45S rRNA is performed by RNA polymerase 1 and processed in 90S pre-ribosomal nucleolar complex to give 18S, 28S and 5.8S rRNA. So this is how this cleavage will occur. We are not going to again into the detail, but yes, these type of cleavages will be seen in the RNA, one RNA to give us many small rRNAs. Okay. Now the post-transcription modification in case of the tRNA. How does it happen? So in both eukaryotes and prokaryotes, tRNA processing occurs. It is transcribed as long precursors. Sometimes single primary transcripts carry more than one tRNA segments which are separated by cleavage. So sometimes two tRNA comes into one single transcript which is then further cleaved to make the separate tRNA. So processing of tRNA involves cutting off of extra sequences by endonucleases such as RNase P at 5' end and RNase D in the 3' end. RNase P is a ribozyme with RNA exhibiting catalytic activity. After removal of the sequences from 3' end, CCS sequence is added via enzyme tRNA nucleotidyl transferase. This enzyme binds to CCA sequence at its active site and phosphodiester bond is formed with the 3' end. So CC sequence is important for the recognition of the amino acid and obviously the stabilization of the tRNA. Furthermore, there is a base modification occurring simultaneously such as methylation, deamination, reduction. In case of pseudouridine, uracil is removed and reattached to sugar through C5 end. Okay. So this was the RNAs. Now the next thing that we are going to discuss about is the different genes that are different kind of operon models that are present in bacteria. What are these operons and how they are important? Let's see. So typical bacterial cell contains several thousands of gel and some genes carry out universal tasks and are constantly active called as the housekeeping genes. So genes that are constantly active, they won't be shut at any cost till the cell is alive, are called as the housekeeping genes. So the examples of housekeeping genes include those that facilitate the synthesis of protein and rRNAs. The majority of genes, however, only become active when their byproducts are needed. Such genes should not be expressed constitutively because that energy could be used for more productive tasks such as gene expression is controlled. Such genes expression is controlled, so their products are only produced when and where they are required and in accordance with the needs of the cell. The phases at which a gene expression can be controlled are numerous. There are numerous phases for the control of the gene. You will see further. You must have heard in the when sir was explaining about the operon models. ठीक है. So in prokaryotes, the most common step at which the regulation of a gene expression occurs is transcription initiation. It is energetically the most efficient step to regulate gene expression. The transcription regulation occurs in steps after initiation, specifically during elongation and termination. Prokaryotic transcriptional regulation is accomplished by gene regulatory proteins that bind with the regulatory sequences near the transcription start site of a transcription unit. Gene regulatory proteins, the products of regulatory gene, are of two types. Either they will be activator, either they will be repressor. That you must have learned when you were reading about the operon models. So the basic concept of how gene regulation occurs at the transcription level in bacteria was provided by the classical model called as the operon model. This was the model which was formulated by Jacob and Monod in 1961, and it was one of the best way to understand the transcription level in bacteria for different kind of the genes. Either there will be activator, they will be repressor, or what. So what is an operon? An operon is the regulatory system mostly seen in prokaryotes and bacteriophages, in which a group of structural genes are transcribed together under the control of a single promoter. So the promoter here is single, and as I've told you, the transcriptional unit in case of prokaryotes is polycistronic. So the transcription will happen for all of the genes that are present in the single structural gene and in the whole group, and the promoter will be single for all of them. Generally, operons are very common in prokaryotes and bacteriophages, but it's also found in some eukaryotes. The main difference is that the expression of prokaryotic operon leads to the polycistronic mRNAs, while eukaryotic operon leads to the monocistronic mRNA, because in eukaryotes alternative splicing can happen, but it is generally the single gene for single protein. General structure of an operon: it will be having a promoter, it will be having an operator, then it will be having the structural gene. And this was the very basic structure of any operon model. So the regulatory gene codes for a protein called as the regulatory protein, sorry, which either acts as a repressor or activator, which controls the operon, but it is not a part of the operons because it has its own promoter. Okay. So regulatory gene has its own promoter. Now, what is a promoter? Promoter is a nucleotide sequence that acts as a site for RNA polymerase attachment and transcribe a gene. RNA polymerase recognizes the promoter and then initiates the transcription. Okay. So the operator is a segment of DNA that acts as a site of repressor attachment. It is generally situated in between the promoter and the structural genes of the operon. And the structural genes are at the end of the operon has a stretch of structural genes, a set of structural genes. These genes are transcribed into a single mRNA and finally translated into the proteins, which for which all of this transcription and everything is going on. Now the regulation of an operon, it can be a negative control, it can be a positive control depending upon what kind of an operon we are dealing with. There are two types of transcriptional controls. The negative control, in which regulatory protein acts as a repressor which binds to DNA and inhibiting the transcription, while in case of positive control, in which the regulatory protein acts as an activator which stimulates the transcription. So it is that either the stimulation or the repression in the presence of a regulatory protein is what makes it either an activated operon or a repressible operon. Sorry. Operons can also be either inducible or repressible, as I've told you. Inducible operons are those in which transcription is normally off, not taking place, and it needs inducer to induce the transcription or turn it on. While in case of the repressible operons are those in which transcription is normally on, but something must happen to repress the transcription or turn it off. Okay. So it needs molecule to turn it off, while the positive one will need the molecule to turn it on. Okay. So a negative inducible operon, negative repressible operon. पॉजिटिव इंड्यूसिबल, पॉजिटिव रिप्रेसिबल। नेगेटिव कंट्रोल आप लोग को बता दिया, इंड्यूसिबल, अनइंड्यूसिबल या रिप्रेसिबल, वी हैव डिस्कस्ड इनफ. Now, the characteristic is the definition generally on but can be turned off by repressor in case of repressible operon, in inducible generally off but can be turned on by inducer. Relation to metabolism, repressible is anabolic and inducible is catabolic. Repressor protein produced as inactive form while the inducible operon is formed as the active form. ठीक है. Example, for example, in case of repressible operon, it is the tryptophan operon, while in case of the lac operon, while in case of the inducible one, it is the lac operon. ठीक है. What is the lac operon? So the lac operon of an E. coli contains genes involved in lactose metabolism. It's expressed only when lactose is present and glucose is absent. This is something you guys must remember always. कि while we are studying a lactose operon, then it should be kept in mind always कि when glucose is absent and when lactose is present, then only the lac operon will work. Why so? We'll just study it in detail also, but just for your knowledge to keep in mind two simple points. If glucose is present, then the bacteria won't use any other sugar source at its for its energy. So obviously glucose must be absent so that it can use lactose as its energy source. And to use lactose as its energy source, it must have those enzymes which will help uh, in the usage of lactose, or we can say in digesting the lactose so that it can be used as an energy source. So this was a general thing to remember, but in detail we'll see what are the factors involved which helps in the actual activation of the lactose operon in the absence of the glucose, and why absence of glucose is important for the activation of this lac operon. Okay. So lactose can be broken down by E. coli, but it is not their preferred energy source, as I have told you already. They would much instead use glucose if it were available. Lactose can be broken down more slowly with less energy than glucose. However, if lactose is the only sugar source present, then E. coli will immediately use it as a fuel. So it will depend upon the absence of the glucose only. Okay, so the lac operon contains three structural genes: the lac Z, the lac Y, the lac A. These genes are transcribed as single mRNA under control of one promoter. ठीक है, so the promoter will be single, as happens in case of as happens in case of, एक मिनट, दिख रहा ना आप सबको? Yes. So as happens in case of in case of the prokaryotes, thus it is a polycistronic transcription unit. So it will have three structural genes here with single promoters, the same promoter for all of these three genes. So the lac operon is typically shut off, repressed in normal condition, but can be activated in presence of the inducer lactose, or we can say allolactose also, which functions similar to lactose. Thus lac operon referred to as the inducible operon, because when the lactose will be present, then it will be induced and the genes will be formed, the RNA will be formed, and then that RNA will lead to the formation of the protein. So it is inducible type of operon. So this is the information about the structural genes of the lac operon, ki which gene performs which function. So the structural genes are lac Z, lac Y, and lac A, which are actually help in the formation of these particular enzymes: beta galactosidase, beta galactoside permease, and beta galactoside transacetylase. So these three enzymes are important for the consumption of lactose as a sugar source. Then the regulatory DNA sequence involves the promoter, operator, and cap binding site, which are important for the binding of this RNA polymerase and then start the transcription process. And regulatory gene here is the lac I. ठीक है? This is also called as the inducible. So when lactose is not available, The lac repressor strongly binds with the operator and stops RNA polymerase from initiation transcription. However, the lac repressor loses its capacity to bind DNA when lactose is present, and it leaves the operator and floats away. So this is the case we are reading about when lactose is not available. Okay. So repressor will obviously strongly bind to the operator and stop the RNA polymerase from moving forward. And the lac repressor loses its capacity to bind DNA when lactose is uh, however, the lac repressor loses its capacity to bind DNA when lactose is present. It leaves the operator and floats away, making it possible for RNA polymerase to transcribe the operons. So, this was this a comparison what happens in case of the absence of the lactose and in the presence of the lactose. When lactose or allolactose, which is an isomer of lactose, is available, so allolactose binds with the repressor, which changes its shape and prevents it from binding on operator, so that RNA polymerase can easily bind with the promoter. Thus allolactose acts as an inducer. So allolactose is an inducer of this lac operon along with the lactose, because allolactose is an isomer of lactose. Some other lactose analogues are isopropyl beta D1, thiogalactopyranoside (IPTG), phenyl beta D galactose, and thiomethyl galactoside (TMG). So overall condition, what happens is, in the absence of the glucose, when lactose is present, strongest transcription of the lac operon occurs. The lac repressor cannot bind with operator because the inducer is present, and the CAMP levels are also high because glucose is absent. So activated cap helps the RNA polymerase to bind with the promoter very strongly, permitting the high levels of transcription, while there is no such thing happens when both of the things are absent, the glucose and the lactose, both of them are absent. So if we'll go into the summary of the lac operon, what will be the summary? The lac operon contains genes that are not involved in that are involved in lactose metabolism. Lac operon is negative inducible operon. The genes are expressed only when lactose is present and glucose is absent. The operon is turned off in normal condition. The operon is turned on and off depends upon the glucose and the lactose level, because if glucose will be present, then bacteria won't use the lactose part. Okay. It won't bind to the lactose part, so that is why this happens. The lac repressor blocks the transcription of the operon by binding with the operator, and in the presence of lactose it stop acting as repressor. And catabolite activator protein acts as an enhancer, activates the transcription of operon, but only when glucose levels are low. So this also depends majorly on the levels of the glucose inside a cell. Okay. uh, this was just uh, an indication to what happens in which condition. Okay. Now we'll talk about the tryptophan operon. So the tryptophan operon found in E. coli bacteria, it is a group of genes that encodes enzyme for tryptophan biosynthesis. So in case of lac operon, the genes involved are for the metabolism of the lactose, while in case of the tryptophan, the genes involved are the genes for the biosynthesis of the tryptophan. So it is an anabolic or negatively controlled operon. It always remains on in normal condition and off when the tryptophan levels is high. The tryptophan itself acts as a repressor here. One unique feature of the tryptophan operon is in attenuation. Like regulation by the tryptophan repressor, attenuation is a mechanism for reducing the expression of tryptophan operon when levels of tryptophan are high. However, rather than blocking the initiation of the transcription, attenuation prevents the completion of the transcription. So it won't stop the initiation. Initiation will happen, but it won't let it complete. It won't let it get completed. Just a moment, guys. Next is So tryptophan regulation in absence of tryptophan या low tryptophan, when there is little tryptophan or absence of tryptophan in the cell. In this condition, the tryptophan repressor is inactive because there is no tryptophan available to bind with the repressor and activate it by conformational change. So inactive repressor cannot bind to DNA, and that is operator or block transcription, which allows the tryptophan operon to be transcribed by RNA polymerase. So in absence of tryptophan, it will keep on transcribing, but when tryptophan is present, it will shut off. In presence of tryptophan, what will happen when there is a high level of the tryptophan or presence of tryptophan in the cell? Tryptophan acts as a compressor and converts the inactive repressor aporepressor into the active repressor holorepressor by inducing a conformational change on it, and thus holorepressor binds to the operator and blocks the attachment of RNA polymerase to the promoter. This shuts off the transcription of this RNA of the structural gene. So in absence of tryptophan, it will be shut off. In absence of tryptophan, it will be on, and in presence of tryptophan, it will be off. So it is a repressible type of the operon. Okay? So there are major differences in both of these operons. Next is the arabinose operon. So arabinose operon, the 5 carbon sugar L-arabinose must be broken down by an operon as the L-arabinose operon, also known as the ARA or ARA-bad operon. In E. coli, the 3 structural genes Arab A, Arab B, Arab A, and Arab D, together known as Arab-bad, are found in the L-arabinose operon, code for 3 metabolic enzymes needed to break down L-arabinose. These genes generate the enzymes ARA-B, ARA-A, and ARA-D, which catalyzes the conversion of L-arabinose to D-xylulose 5-phosphate and intermediate in the pentose phosphate pathway. If you haven't read about pentose phosphate pathway, we may read it in the further lectures. Otherwise, you can read it from any biology book of either plus 2 class or above. You will find this there, pentose phosphate pathway. A single transcript mRNA is produced from the transcription of the structural gene in L-arabinose operon. The catabolite activator protein CAP or CAMP complex, which is produced by the regulatory gene ARA-C, regulates the expression of L-arabinose operon as a whole. So there are also many genes involved in arabinose operon. In addition to being under the control of CAP-CMP activator, the L-arabinose system is also positively or negatively regulated by the binding of ARA-C protein. ARA-C performs as an homodimer and interact with the operator initiator region of arabinose operon to control the transcription of ARA-bad. A DNA binding domain and a dimerization domain make up each ARA-C monomer 2 domains. And the binding of arabinose is carried out by the dimerization domain upon binding to arabinose. So ARA-C undergoes a conformational shifts and adopts 2 different conformations, and the binding of allosteric inducer arabinose is the only factor that affects the conformation. And when the concentration of RSC rises too high, RSC can potentially advert order regular own expression, and by attaching dimer RSC to the operator region, RSC production is inhibited. So basically, it performs the regulatory function. So this was all about all of the operons. I hope you must have read it very clearly from the lectures that sir had provided. It was just an overview. This was very, very superficial overview. We didn't went in the details. You can learn it from the videos. Still, you have any kind of confusion, you can ask me. Now we should move towards the assignment questions. So as our practice goes, I'll ask you a question. You will write the answers in the chat box so that everyone can perform and everyone can participate. Okay. So yes, the first question here we have is, during transcription only one strand is transcribed in so that the transcribed sequence is identical with one strand of DNA, known as what? Chat box, everyone. I would love to see your answers on that. Okay, so the answer to your question is coding or sense strand. Yes, so yes, you are right because we have read it today only that it is the coding or sense strand to which the transcribed sequence is exactly similar. ठीक है. Next question is Rho protein is an hexamer ATP dependent helicase which involves in Rho dependent what? Bhavna, I think you need to check your answer again. Read the question again, Bhavna, and then answer, please. Yes, now it's correct. So the answer to this question is termination of transcription in prokaryotes. Next question. During post-transcriptional modification, splicing mechanism of both group 1 and group 2 involve similar steps of two following reactions. What are those reactions? Yes, it is the transesterification reactions. Okay. Yes, it is the correct answer. Next question. Sex determination in Drosophila is the result of following during post-transcriptional modification. Sex determination in Drosophila is the result of following during post-transcriptional modification. What is that? Yes, it is the regulated alternative splicing. In prokaryotic operon system, the expression of prokaryotic operon leads to what? Yes, it is the polycistronic mRNAs. Okay. Next question. The repressor or activator which controls the operon system is not a part of operon because it has its own promoter. Yes. Next question. Operator is a following and acts as a site of repressor attachment. Yes, correct. It is a segment of DNA which acts as a site for repressor attachment. Badhiya. Next question is the lac operon contains three structural genes. What are those genes? Yes, lac Z, lac Y, lac A. Galactosidase, transacetylase और तीसरा क्या था? Whatever it was, you guys must have read. Next question. When lactose is not available, the lac repressor following with the operator of the lac operon. The lac repressor dash with the operator of the lac operon. When lactose is not available. Yes, it strongly binds with the repressor. ठीक है? Next question. In lac operon, following acts as inducer. What is that? Allolactose is the one which acts as an inducer. High amount of lac operon transcriptions are only possible without what? Yes, it is D, with in the absence of glucose. In the absence of glucose, the high amount of lac operon transcription can occur. Okay, next question is tryptophan operon is dash when tryptophan concentration is high in the environment. What happens to the tryptophan operon? Question number 12. Repressed. Are you sure, guys? I think repressed is the one when is attached. Now it is also inactive, so yes, both the answers are correct, but the appropriate term will be inactive. Okay, so the appropriate term for this is inactive. Okay, next question. In the tryptophan operon, attenuation means, question number 13. Attenuation leads to what? Prevents the completion of transcription. Okay, so it is the prevents the completion of transcription. The three structural genes ERA B, ERA A, and ERA D are found in what? Very clear from the options, from the name also. Yes, it is ERA bad operon. ठीक है. Question number 15. The L-arabinose system is positively or negatively regulated by the binding of the following. Question number 15, everyone. Yes, it is ERA C protein. ठीक है. So with this we have come to an end of today's lecture. If anyone wants to ask any question, they can ask me. Because we have discussed the assignment, we have just had an overview of what we read in the last week. Anything else, if you guys want to ask, you can ask me. Any questions, anyone? You can take 2 minutes if you want to ask some questions. I'm waiting. Any questions, anyone? No questions? Okay, then. If you guys don't have any questions, you guys can leave the meeting. Happy Holi to everyone. Enjoy your Holi and have a very safe Holi. Okay? You guys can leave the meeting now. You're welcome, Arpi. Sorry if I pronounce your name wrong. You're welcome, everyone. Have a very safe and sound Holi. Enjoy. Now you guys can leave the meeting.